Welcome, DCRC Labs welcomes Dr. Kushwant Yadav, sir. And sir, we welcome you and very good morning. Sir has an experience of more than 19 years in both industry and academics. He has been a research scientist in Claris Life Sciences, Ahmedabad and Alembic Limited, Vadodara. Sir has completing AICT RPS project as principal investigator with a grant of rupees 18 lakhs. He has done collaborative research with National Institute of Research for research in reproductive health, ICMR lab, Mumbai, and Institute of Nuclear Medicine Allied Sciences in Mass DRDO Lab, Delhi. His publication has been listed in 25 hottest articles by elsewhere, most read articles by Parenteral Drug Association USA, most view articles top 10 by AAPS USA. He is recipient of Fellow of Institute of Chemist India. Recently, Dr. Kushwant Yadav, sir, has been included in the Stanford University elsewhere world ranking of top 2% scientist 2023 database. His interest area included has nanomedicine, drug delivery, formulation, development of anti-cancer drugs, novel drug delivery systems for glaucoma, neurodegenerative diseases, microparticles, polymer-based kinetics. Beside this, sir has completed his postdoctoral fellowship by joining postdoctoral research associate at the University of Oklahoma Health Sciences Center, US in 2015, and he also the recipient of Young Achiever Award 2015 by AAP Telangana State Branch. Third into US conference, Hyderabad, 26th September 2015. Sir has also editorial board of various reputed journals like DARU, Journal of Pharmaceutical Sciences, Springer, which having the impact factor of 1.654 since 2015. He is also associate editor board members from Drug Delivery Letters, Bentham Sciences, and Associate Editor Board member of Nano Science and Nanotechnology Asia, Bentham Science. Besides, sir has vast years of experience in the field of nanotechnology. We all welcome you, sir, for today's topic on nanoparticle delivery systems for targeted cancer therapy and imaging. Thank you so much, sir, for giving us your valuable time and discussing the topic with the audience of Drug Chemistry Research Center. Now, over to you, sir.
Thank you, Ashutosh. I would want to thank you for inviting me. It's been a long time. I'm happy, and I'm so happy to see the progress that you're doing, and you're helping so many people with your internet programs and the other offline things that you're doing, which I just heard. I'm very happy for that. I'll share my screen.
Yeah, definitely, sir.
Is my screen visible?
Yes, sir.
Okay. The title of my talk, as you all know, is Nanoparticle Delivery Systems for both the cancer therapy and imaging. And since I'm the third speaker, I'm happy that the platform is already set. You're all well aware that what is going on, and I'm sure will be much more easier for me to now take it ahead. Now, to begin with, when you look at this point, that because we have been talking about nano drug delivery systems, and then we talk about whether are they really feasible. So my first slide itself says that, of course, hugely yes. It's been a long time, been a long journey. So these are two recent news from two different areas. The first one talks about is the use of nanoparticles to aid the cancer treatment. And it's very clearly understood that these nano materials will be, trumping the traditional drugs with their good biocompatibility and non-toxicity nature, they will be way ahead of the conventional drug delivery system. And the second news again gives an information on the diagnosis part, where the liquid biopsies for the cancer, which has been a troublesome task, can be really done with the use of nanotechnology. Both the users in 2022 and 23. Now, we are all aware what nanotechnology is, and we are aware of the nanoscale, which is 1 to 1000 nanometer in size. But let us look at what the NIH talks about. What do they feel is nanomedicine? So let me bring to your point of interest. The word nanomedicine takes care of highly specific medical intervention. Now, this is exactly at the molecular scale, where we can be utilizing it for diagnosis, prevention, and treatment of diseases. To compare the sizes, I have put right in front of you the size of hemoglobin, and you'll see here that hemoglobin, as an example, could be somewhere close to 5 to 7 nanometer, and the antibodies could be 12 nanometers. A very good example to compare with is also the influenza virus, which is also closer to 100 nanometer. Now, our nano drug delivery systems, again ranging from a 10 to 500 nanometers, are beautifully used as drug delivery vehicles, because anything that is close to 50 nanometer can enter most of the cells, and most of them closer to 20 can be circulating throughout the body. If you still ask me, why do we need nanomedicine? I will tell you that strongly, yes, we really need nanomedicine. And this was told to us way back in 1900s by the scientist Paul Ulrich, who said, this is the concept of magic bullet. And this concept of magic bullet only works on nanoscale. Some of the pictures over here tells you how this is the modified nanoparticle drug delivery systems, along with the brush-like structures, which is ensuring that the nanoparticle remains in the blood circulation and it reaches right in the place where it is meant for. A small GIF image which tells you how easily the nanoparticle is able to travel through. This is the doxorubicin bound nanoparticle going into the bloodstream and entering into the tumor microenvironment. So what makes nanomedicine different is that they are able to get inside the cell. And once they reach inside the cell, of course, they are able to damage the cell completely. They are able to assist in so many different biological processes, such as to assist the damaged intracellular structures. And sometimes one might not even imagine they are able to replicate themselves, correct any of the issues, that is, the gene structures, even at the level of DNA molecules, is also possible. One more example is recently the Purdue University in the US have said that nanoparticles are able to exit from the blood circulation and enter very quickly into the tumor microenvironment, which is able to enhance the better treatment of cancer. They've also had the procedures for the breast cancer. They have developed a melanoma model. And because of these two examples, where they have worked with polyol modified nanoparticles, they are able to report that the cancerous cells can themselves be located, the tumors can be located, and the nano drug delivery systems can be used to reach those places directly. So every time when we talk about cancer and when we talk about the nano drug delivery system, we would say that I would want my delivery system to be directing the drug to its target site. Not to make it more tough and complicated, here I would want both the things. My nano drug as a nano carrier, as a nanoparticle, would itself also be interested to be reaching the tumor microenvironment, as well as once it reaches there, it should be able to deliver the drug and allow the drug to act the way it is actually meant to be. So eventually what will happen is the nano medicine on a whole is improving my drug's efficacy, reducing the toxicity, and because it is able to work in both the fashions of temporal and spatial targeting, it is able to stay there for the sufficient period of time. Just for an example, because we are talking about cancer, can we really just think that do we really need nano delivery systems or nanotechnologies intervention? The answer again is yes, because the conventional chemotherapy eventually fails. When we talk about the cancer cells, we are well aware that the growth of the cancer cells is much, much more faster as compared to the other healthy cells. And because of which, eventually the total number of doses keeps on increasing. These bolus doses are intense in nature, has got serious side effects. At some point of time, the patient might even discontinue the therapy. And even if they continue the therapy, still what will happen is that more and more of chemotherapy, at some point of time, the radiation, which is there required to kill the tumor cells, eventually kills the patient. It's very painful if I say the treatment is not that effective in killing the cancer, but eventually the cancer kills the patient. And this is the reason which brings us closer to an alternative nanomedicine treatment. Again, going back to what happens at the growth of the tumors. So eventually, because they are, even if they are single, they would replicate. Once these small in number cancerous cells are there, they would take off the entire nutrient supply which is available close to them. Eventually, all of the healthy tissues which are nearby can never be able to compete. So the healthy cells will not be able to withstand the cancerous cells. And after some time, these tumor cells would be displacing off all the healthy cells. A stage like the diffusion limited maximal size point comes, and after some time, these cells do not initiate apoptosis. I hope if you are aware, if they do not initiate apoptosis, there will be no programmed cell death. One more wonderful thing in the end happens is these tumor cells, even when there is no nutrient available, because after some time, if there is no good nutrient available, they usually metasize and they are still able to divide. So this is the picture which gives you the information. The first stage where I was talking about the diffusion limited maximal size, it reaches, and then this wrong thing known as the angiogenic switch occurs. Because of this angiogenic switch here, you see here, there is an imbalance of both the positive to negative regulators, and because of which the endothelial cell proliferation and migration happens. And this endothelial cells eventually starts extending towards the tumor cells. You see here, right, starting from B to C, and eventually at the point of D, you see here, it's a fully vascularized tumor. And it has got a huge metastatic potential. That is what is the most painful. That is what when we say the cancer has reached the fourth level. Now, once any cancer which has become metastatic, it will be very difficult to then kill those cancer cells, because of these blood vessels, they will be traveling throughout the body in a very easier way. Again, this is the colorful picture, gives you the information on the classical angiogenic switch. You see here, in the start when the blood vessel was so less, so small, and then the tumor vasculature is increasing in size, and eventually you see a huge growth of angiogenesis, and all over the place, these cancer cells are able to migrate. So you see here where it started and where it led, and again you see that it is able to join two vasculature tissues, connecting them and spreading the cancer everywhere in the body. If you ask me, can I connect the cancer as a drug delivery target? I would say that it is one of the most challenging and is actually the most interesting delivery systems, because cancer in itself gives us so much different variations and opportunities which actually can be exploited to be used as a drug delivery target. So this slide gives you information on cancer itself has got so many opportunities which we should be able to utilize. One of them is the tumor blood vessel has got several abnormalities which are way different from the physiological vessels, and they have a discontinued endothelium which also has got gaps between the cells. And this gap, again, I'm trying to bring to your kind information that this gap, the broken endothelium, discontinued endothelium, the abnormal endothelium has got pore sizes closer to 100 nanometers to several hundred nanometers. And that is the reason we have to continuously say that it's not the conventional drug delivery system. Because when my students ask me that, suppose if I give my delivery by parenteral route, suppose I give an intravenous injection. Now, the drug is in an intravenous injection form is much, much more smaller in size as compared to nano, that what I've been talking about. But the difference between the two is my this molecular level conventional delivery is definitely not able to enter these gaps, or is not able to exploit the discontinued endothelium. Another important point here is the two important ways in which nano itself is able to exploit. For example, the transport pathways available in these open gaps is two. The two are the interendothelial junctions and the transendothelial channels. Because of both of these, it's very easy for the nano carrier to reach, whereas the others are still struggling. Just in brief, a huge important applications of nanoparticles. When I talk about is a good selectivity, is that nano ensures that the drug reaches the right place and is selectively able to destroy the cancer cell. When I say they are having reduced toxicity, what I mean is that because nano doesn't go here and there, is not able to enter into more and more of organs and tissues, hence, because of not coming in contact with the normal cells, the toxicity is also reduced. It's very well able to act at the right cellular level, because in the next few slides, I'll show you the four different levels of targeting. So this is the cellular level at which nano delivery can be working. Efficacy, not to be talking about, we already know a huge efficacy improvement is there as compared to the conventional delivery. And why this is possible is because the processes of endocytosis and phagocytosis, which is difficult for the normal cells to do, the cancer cells are able to endocyte the nano carrier, which is having the anti-cancer drug. They are able to encapsulate most of the drugs, whether you talk about the drugs being hydrophilic, hydrophobic, the drug being macromolecular in size, some of the peptides, protein-based drugs, all of them, everything can be accommodated in nano-sized drug delivery systems. For the oral delivery, where we might be thinking, for example, doxorubicin gets destroyed in the GI tract, so cannot be given by the oral route. But then this premature degradation can always be avoided by use of nanocarriers. Greater enhanced absorption and reaching at the right selected tissue is one beautiful thought about the nano particles. Pharmacokinetic profiling is improved, distribution profiling is improved. I would say, I would give an example of this drug, temozolomide, if you know, is being used for the brain cancer. And half of you would be shocked when I tell you that if you talk about the bioavailability of temozolomide, which people would believe is really good, what is reported is maybe closer to 80%. But if you ask me, that 80% of the bioavailability is in the blood circulation, not in the brain. So that disconnect of where the drug has to be reaching. So the impermeability of temozolomide for the brain itself is an issue, where you talk about brain cancers and people dying as early as within a year, is the reason that we do not have good control over the pharmacokinetic and distribution profiles of these drugs. And the last point here says is a greater extensions of the half life and improved shelf life of the product, which is again possible by nano drug delivery systems. Targeting is again a beautiful thing that is possible only because of nano drug delivery system. One easy way is the passive targeting, because I just told you about the enhanced permeability and retention effect, which is only because there is a hypervasculature, there is a defective vasculature architecture at the site of cancer, and because of which there is a deficient lymphatic drainage. And this issues can be very well utilized by passive targeting, where we do not have to struggle much. For example, if I work on my plain PLG nanoparticles, or even if I coat them with MPEG, I might be able to reach the nanocyte, the cancerous sites. Whereas if I have to target them actively, then I have to be working on my ligands. Before I go to active, just a picture of passive tissue targeting. You see here, so these are my nanoparticles, they are traveling. And you see here, these are the gaps that I was talking about. You see here, in between these endothelial cells. And these gaps can be very well utilized, and because of which you see these nanoparticles entering into the tumor microenvironment and are able to attack. Whereas in the opposite, in the downside, where you see, because where the normal conventional therapy is not able to reach quickly, there will be a development of angiogenic vessel, and there will be an ineffective lymphatic drainage, again causing issues. Now, coming to active targeting, because most of the active ligands, for example, folic acid. Now, folic acid will specifically be targeting to whatever receptor is there within the cancerous cell, and it will be able to reach the right site. The first point is the conventional, where just due to its size, I would say just because of utilizing the RES system, just because of the correct sizes of maybe between 20 to 200 nm. And if I am able to surface modify them by making use of the pegylation, I will be able to make them long circulating also. This is the picture of active targeting, where you see that I can be making use of any of the biological ligands. Now, these biological ligands in the form of another nano, a protein, a peptide, aptamers, different small molecules, all of these can be attached. And once these active targeting are able to recognize the receptors which are rightly available at the disease sites, it's very easy for the ligand receptor type interaction to happen, and because of which the carrier is able to rely on the biological interfaces between the target cells and is able to reach the right side. These are some beautiful examples. So as a receptor, I have folate, I have transferrin, EGFR, HER2 receptors for the breast cancer, and the different series, for example, mainly the CD44, where I have my targeting moieties. I'll use folic acid to target folate. I'll use my different anti-EGFRs to target EGFRs, different drugs like cetuximab, or the different HER2 receptor-based for breast cancers, the HA targeting for reaching CD44. And this is where different cancer cells can be actively targeted. So where I'm using my antibodies, fragments, proteins, lipoproteins, even hormones, also utilizing the correct use of small and higher low molecular weight ligands. Just for an example, divergent, other than the active and passive, I have my physical targeting. I can be making use of magnet, ultrasound, hyperthermia, the phototherapy. This is just an example where I'm showing you the ultrasound waves, which can be used to enhance the delivery of my nanoparticles, electrical waves, light therapy. This is more information on hypothermia. For what happens with hyperthermia is, a small increase in temperature, roughly reaching to 43 at the local site. What it does is vasodilation. Now, because of this vasodilation, there is an enhanced tumor perfusion. And because of this enhanced tumor perfusion, the drug delivery is improved. The heat activable vehicles are able to reach the target site. Going back, chemical targeting with using different enzymes, different chemical reactions, the biological targeting, which makes use of different antibodies, peptides, gene expressions. So, biological targeting and physical targeting helps us in reaching the right site. I was telling you, I'll take you to the four different orders. So, just taking liver as an example, the first level of targeting is my organ level. If I go right more inside it, I would reach the hepatocytes, which I could call them as my second order cellular targeting, reaching much more inside my different intracellular levels like mitochondria or nucleus. I would call them as my third order targeting. Finally, the fourth one of the most complicated, where I would say I would be targeting the F1 subunit. Imagine, to that extreme level is also possible. The specific molecule within the cell, just like the F1 subunits which are available in the mitochondria, even that we are able to target, and that is because of use of nano drug delivery system, which is able to exploit all of these force. But some more terms, if you are aware of the use of double targeting, where we are making use of both the temporal as well as the spatial placements of the drug, a combination where I'm also using my homing devices, which are being used to navigate the right place, in addition to the molecular specificity of my product, gives me a beautiful combination delivery, a dual targeting with my carrier molecule and my therapeutic activity. And inverse targeting, for your information, is the word where I would initially be giving blank colloidal carrier injection. For example, because most of the conventional nanoparticles would quickly reach the RES, the reticuloendothelial system, and will be taken care by the MPS organs, the liver and the spleen. So because I don't want my nanoparticles to be going there, initially I have to block them, and I can be blocking them by making use of the inverse targeting. Some examples of the different nanocarriers, not going that much in deep, just for some names, I can be using other than the polymeric nanoparticles, my lipids for liposomes, solid lipid nanoparticles, stealth liposomes, stealth technique, again making use of MPAC, different drug conjugates, polymer protein conjugates, inorganic nanoparticles like the metal nanoparticles, and so many of them. A picture of liposomes, beautiful, because here I can be exploiting both hydrophilic and lipophilic characteristic features and ensure that both types of drugs, and again make use of different lipids to ensure good targeting. Dendrimers, which as you can see the picture, are highly branched macromolecule structure, and because of these highly branched, and I can be really increasing it to the fourth generation also, where I can be improving my targeting effectiveness. Micelles or reverse micelles, where beautifully they are able to self-assemble, make use of the amphiphilic block copolymers, really much more smaller in size, 5 to 50 nanometers, and are beautifully able to be making or exploiting different copolymers, different block copolymers, and because of which they are able to reach the different sites, and because of different types of the bondings that they might have, they may be working by the enzymatic degradation. For example, if I make my micelles pH sensitive, what I can be achieving here is at the pH of the tumor microenvironment, which we know is much more acidic than the normal body pH. If my blood pH is 7.4, my tumor microenvironment is just a 5.5. Because of this drastic change in pH, I can be exploiting micelles. I can be making my micelles pH sensitive, and because of that pH sensitivity, one is there can be a quick shift in the charge in the surface potential, maybe a cationic to anionic switch, and at that point of time, what will happen is I will be having my drug delivery system reaching at the site, and after reaching this site, it will be able to even release the drug at the right place. This is for examples from our own labs. One example where we tell you, we show it very clearly. So if anyone still feels that whatever we are talking is just in the labs? No, it's actually possible. It is practically visible right in front of you, where I tell you this docetaxel targeted nanoparticles, the way they are compared with untargeted, and where plain drug is being used, and this poor mouse where this was left untreated. You can see there is a huge drastic change in the tumor mass. So the first one where docetaxel targeted nanoparticles have been used, the size of the tumor has been drastically reduced. And on top of it, the most important thing is there was 100% achievement in treatment, whereas in the other cases, just with pure drug docetaxel, only 14% of the mice survived. Again, one more example from my own lab here, where we have shown that the nanoparticles are able to remain in the blood circulation for a much more longer duration of time. You see here is this purple color over here is technetium TC99 coated so as to get the amount of drug reaching. The study was done at the INMAS lab, DRDO lab in Delhi. Here you see these pegylated nanoparticles are able to be remaining in the blood circulation in a not only in a huge quantity for a longer duration of time, whereas the conventional ones and the plain drug is over within 1 to 2 hours. Similarly, when you compare the effectiveness of each, so for killing 50% of the cells for my plain drug, this is etoposide, if I need 18 micromolars, where my nano drug delivery system, so these are three different nano formulations of the drug. You see, each one of them is much more lower than a 6 micromolar, which means I can reduce 1/3. And then you ask me, why would there be less toxicity? And the answer is right in front of you, because I have reduced my dose to 1/3. So where I was needing an 18 micromolar concentration to kill 50% cells, I just need a 6, and maybe even much more less for my MPEG nanoparticle, which is just closer to a 5. So a drastic improvement. One more beautiful picture. This is taken at the NIRRH, the ICMR lab in Mumbai, where I did this study. This is again my own research. Here, if you see this, these are the cancer cells. This is for your information, DU145, the prostate cancer cells. And if you can visualize it, this is one individual cell. And this is the cell membrane. The most inner part of this is the nucleus, and this is the cytoplasm. These fluorescent things that you see over here are my pegylated nanoparticles. So these are pictures taken at 4 different time intervals. The first R, you see the drug is in the nano formulation, is able to enter into the cell. In the second R, you see they are roughly placed in both the places, much more available in the cytoplasm, and as well as trying to enter into the nucleus, because in the upper picture, you don't see them entering into the cell nucleus. The most interesting picture here is the last one. At the 4th R, you see here, the nanoparticles are only within the nucleus. You see here. And that is what we talk about. That is what is the correct targeting that is possible. And believe me, this study is not recent, was done some years back. At the time, I just want to share a quick experience. The director at NIRH told, you people keep talking about nano and like nothing happens. What is there? Does it really work? So the moment I showed Dr. C.P. Puri, the director then at the ICMR lab in Mumbai, that see this, my nano drug delivery reaching the nucleus, where my conventional delivery could not even touch the cell. You forget about the nucleus. He quickly, got up from his chair and was looking at the pictures and then appreciating the work and appreciating nanotechnology, that yes, nano has the strength to reach the right site and the right target place. One more picture. This is the Z-stack confocal microscopy, which again gives you an information that if you tell me, is my drug just getting adsorbed on the cancer cell, I would say no, because this Z-stack confocal microscopy gives you a clear picture that it's available right inside the cancer cell. One more of my works which shows you the biodistribution pattern. And here I would want to emphasize on the fact that once we talk on nano deliveries and cancer, what is the major problem is that the drug would eventually be reaching the RES organs. You cannot stop it. And that is where our task of rightly preparing the right kind of delivery systems, which will also ensure that the drug is not reaching liver, spleen, and lungs. So that is where when you talk about your nano drug delivery system, you see here is they are highly negligible. The last part of it you see here, where my conventional drug would definitely go. There's no chance that they would not go, but I'm able to safeguard my nanoparticles from reaching these three organs, because reaching these three organs means the drug is not available to the right site and is going to be going off. And this picture, which gives you the information that nano carriers biodistribution pattern is so beautiful that it is able to reach the places which I would have never reached or which I would have never thought about. Now, here I show you etoposide trying to reach brain. It is negligible. Or if it is trying to touch upon my bone, so especially this was the study we were doing for the blood cancer, where I would want my drug to be reaching the bones and where it's not possible. And here you see my nano carriers are able to reach in a good quantity and a huge percentage comparison of the injected dose. If I talk about reaching both the brain and the bone, that is what the biodistribution patterns of the nano drug delivery systems is beautiful. And to top it all, so even if you say that the nano may not be working or it's just in the books or in research, the answer is again a no, because these are right examples right in front of you. All of these nanoparticles are available in the market. They're all US FDA approved. The first one, I'm sure you must have heard of, Abraxane for sure. Nobody can miss this example of a beautiful nanoparticle albumin bond carrier for paclitaxel. Who cannot forget Depocyst, or the first wonder molecule coming way back in 1995 as Doxil, where we understood that liposomes do wonder and doxorubicin was able to be encapsulated and doing wonders for the ovarian and the breast cancers. And way forward ahead, 2007 to 2012, you can see that there are many, many more liposomal formulations and some polymeric formulations also available in the market. So it is a reality for sure. What we can do, or what else can we do by when I talk about my active targeting? So not only by intravenous route, this is the picture which tells you that the oral route can also be exploited when you talk about the nano drug delivery system. So when I talk about my oral delivery, I am interested in M cells. I am able to target the Pierce patches, which would have been impossible. Otherwise, I would have not even thought about my conventional drug to touch upon the oral delivery and making use of M cells and Pierce patches. I'm able to reach the lymph nodes. I'm able to exploit the capillary walls, extravasation, and I'm able to make use of the movements in the tumors, whether you talk about EPR effect or the tumor region. And of course, as I've already talked, the use of leaky capillary supply at the tumor site can definitely be exploited. So whether you talk about the intravenous or whether you talk about oral, or even in some here examples of topical I have not used, nano deliveries can be actually be utilized. Achha, before I forget, I wanted to tell you I use topical. So if you must have also heard of, the bhasmas. In the traditional Indian story, also the sadhus have been using bhasmas for a long period of time. And let me remind you that bhasmas was nothing but the nano formulations applied topically, and that was the reason it was doing wonders, is because of the nano size, which again gives us the information. If there are some students in the group for their quick information on how you can be preparing nanoparticles. One of the most common methods used is emulsion evaporation, where I would use my polymer and the organic solvent and the drug in one phase, use surfactant and water, because ideally I want to be making the emulsion, so that finally, eventually, I would be evaporating my organic solvent, make use of high pressure homogenization procedure, and I get my beautiful nanoparticles. Another, if it's a water soluble drug, can be difficult for me. So because of which, what I'll do is I'll be using a double emulsion. So ideally a water in oil and water emulsion, eventually again going back to evaporation of the organic solvent, and I get my beautiful nano suspensions. Not to forget, in this, I would quickly centrifuge them so that I remove the unentrapped drug, and then freeze dry them so that they are stable. Another procedure, if you want to make use of salting out approach, where I make use of a salting out agent, make use of uh, solvents like acetone, make oil and water emulsion, and dilute it with a huger amount of water, which ensures that the nanoparticle suspension is being created. One of the most easier, the last method I think that I have here is nano precipitation. So beautiful method here. What happens is I'll be using two solvents, and I'll be ensuring that now these two solvents are miscible with each other. Maybe one good example is making use of water and acetone. You can imagine yourself what happens if I mix acetone and water. They are so friendly with each other. They are hugely miscible with each other. Is that any of my polymer or drug which is available in the water phase, which is available in the acetone, once it comes in contact with water, they'll precipitate out quickly. And depending upon the different sizes that I can be precipitating out in the nano size range. I don't know how many of you are aware that my key area of working is quality by design. So I wanted to quickly touch on the QBD approach also. And when I talk about quality by design being used and combining it with nanotechnology, I love it because here is where I can talk about creativity. I can talk about a work which is distinctive to any other work that is to which it is compared with. And I can ensure the two important characteristic features that I want in my work, that is novelty and quality, almost. A systematic approach, and I don't know how many of you are aware of QBD, but then for us, it's a beautiful process which ensures that predefined ways, my objectives, my understanding of my pharmaceutical development begins, and I ensure that the way I have planned my nano drug delivery systems, it is able to reach my target aim, and my target aim is nothing but the patient. So believe me, QBD only has got one goal, and the goal is patients, clinical safety, outcome, and health for all. These are again some of my nanoparticles to which QBD can always be applied. If you talk about why QBD and why nano, the picture here tells me that all of the physico-chemical characteristic features, which are an inherent characteristic part of nanoparticles, can be done better, can be made more safe in terms of toxicology, can be made biocompatible, and the more important part of where I'm able to work upon the pharmacokinetic and dynamic processes, I make use of QBD. And to end it, when I say I can be working on my scale up, because many of you might say that you can be working for nanoparticles in the lab, but are they scale up? Reproducibility is also there. The answer again is yes. And because I am able to have a huge process control on my processes, I am able to come out with my nanomedicine product. Again, some more examples. I think we've already seen some of them, just a replica of these nano formulations which are available in the market. a quick thought on the role of QBD, because QBD ensures that the nano is efficient. We are able to minimize the risks associated. And to give you a quick recap of why QBD is, because it was way back in 1990s, and now we are in 2023, where ICH itself said in 2005 that QBD should be an intrinsic part of any formulation product development. Although what we understand in nanoparticle is, okay, now in spite of this success, we need to be ensuring that quality by design can only be working if you are able to keep a track on the size, the nanostructure, the composition. And all of this is only possible if you're able to be having a huge impact on the regulatory agencies. Now, QPT, what it does is it's also able to ensure that the regulatory concerns with the scale up, the technology as a whole, and the pharma company, because it comes in contact, it makes it highly coherent process where all of these complex pharmaceuticals are able to come out with the way we have planned. So if you ask me, my designed nanomedicine, I would say is only possible when quality attributes of my nanoparticles come in contact with my quality attributes that I'm expecting in the nano drug delivery systems. And again, a beautiful example which tells me that investments in the last few years has been so drastic in nano that right now, where we started with only 40 products in 2014, I already have more than 100 nanomedicine products rightly approved by the US FDA, and for that matter, even Europeans EMA, continuously working on the growth of nanomedicine and drug delivery systems. Just last one quick thought on QBD, where I'm connecting three things, because I'm always interested in inventions, innovations. Coming in close connect to what I really do not want is imitations. So taking away just the imitation point of view and ensuring QBD ensures innovations and inventions come together, I have to be making sure that QBD, along with CQAs and CMAs, ensure that the quality targets are met. This is the slide which gives you all nano medicine features, right from my size, stability, surface potential, surface decorations, organizations, different stimuli that I can be using. And you can see here how beautifully the design complexity can be made use by QBD concepts. Okay, almost coming to an end. So when I talk about nanomedicine, and when I've shown you that yes, nanomedicine is the way ahead future for me, because even in the previous lecture when you're hearing like ma'am speaking on nanorobots, nanomachines, there will be these artificial antibodies right now, which are working within the RBCs. Just an example of antiviral nanorobots, which can be devised. These diagnostic nano machines, which are able to monitor your internal chemistries in the body, and they are able to reach your nervous system, maybe monitor the pulse, and different things which can be monitored. Everything can be reached. So the pictures here gives you so beautiful information. These, see how quickly you see these artificial antibodies are being attached to it. They are swimming. Another thought which says swimming of the nanorobots, penetrating all of your body fluids, assisting the drug delivery systems, nanomachines doing wonders. Nanorobots was a topic taken in the last session also. Smaller sizes, able to kill the right cancer cells over there. Another picture which tells you these nanoparticles, you see they are loaded with mRNAs. They're able to give you the disease fighting properties of the cells. You can see here how beautifully, which we would have never even imagined. So my nano They can be loaded with the gene therapies, something like my DNAs and RNAs, the mRNAs, and when they reach, they are able to fight. So they make the cell themselves able to fight against all the properties of the cells. Another picture which shows you how beautifully they are able to be there within the blood circulation. And this is an example of the conjugated nanoparticles, where different, maybe you're not gone that deep today, but then these different conjugations, different dynamics of the drug delivery systems, which will definitely improvise the chemo drug delivery systems. You see over here the way it's able to work within the blood circulatory system and is able to reach. This is maybe one last example where these authors have worked beautifully against the triple negative breast cancer, and then they show that it is because of the way this therapy, I mean, this one was a dimeric prodrug system making use of disulfide bonds, as I told you, because this bondings needs to be breaking at the right place. So now here, making use of polyethylene glycols along with the prodrugs and encapsulating the drugs in a way where the prodrug approach ensures that right at the right place of the tumor microenvironment, the drug is able to make advantage of one, that is high drug loading. Second is able to make use of the tumor microenvironments, any responsive behavior. Initially, I talked about the pH responsiveness. We can be making use of temperature. We can be making use of the redox potential. And eventually, we are able to target the triple negative breast cancer, and shows a huge potential for to be effective in breast cancer therapy. One point which we should never miss when we talk about nano is the reversal of multi-drug resistance. And we know that, this, it's a very complicated process, and no one can shy away from the aspect of MDR, and MDR has got issues. There are so many genes, the MDR1, the multi-drug resistance associated protein gene, and I have so many of them, and which has difficulties with my conventional therapy because my drugs at that point of time are not efficient. How do I explain drug effects? Because my all conventional therapy will allow the drug to be thrown out. The cancer cells become so strong. Eventually, they know that it's the drug coming and they throw it out. So now, because I can be making use of the nano drug release system, which will not permit the drug efflux, which will eventually I want cell apoptosis so that the program cell death is possible. Again, a picture which tells you why this MDR would happen and why it will be there is because of these peak glycoproteins are able to recognize the drug, throws it out, makes the efflux process so easy. And that is the reason, because the drug is not able to reach the cytoplasm, and once it reaches, it is thrown off because of endocytosis. The answer to this issue, again, this is again one of our own works where we have shown that nanotechnology provides an alternative approach to avoid the multi-drug resistance. And that is because it's able to encapsulate the drug, safeguard it from any of these efflux procedures, and is able to resist the drug efflux mechanisms. Almost towards an end, let me also tell you, although we have talked so much about good things, but then we should be aware of different challenges associated. So we need to be aware of different delivery systems, different materials, their compatibility, their biodegradation, a clear understanding on their behavior pattern, whether they are bioresponsive, whether they are self-regulatory devices, whether they are mimicking the viruses, whether they are to be implanted within the body. For example, you must be aware of the glidal wafer that is being used for the brain cancer, whether it's a nano chip, whether it's a multi-reservoir drug system, or whether it is taking care of the tissue engineering aspect. So it is not that easy because we are working with so many different polymers and lipids, and these advanced polymeric carrier systems needs to be ensured for their right therapeutic potential. So surely we need to be taking care of the technological challenges also. Almost there, I conclude in saying that nanoparticles are definitely being explored as drug delivery systems for both the diagnosis and treatment of cancer because of the ease in manufacturing, and they are definitely able to encapsulate diverse therapeutic molecules, right from small drug molecules to macromolecules. And that's where I end my talk. Thank you so much.
Thank you so much, sir, for presenting your work. And the slides were very informative and knowledgeable. And first of all, many congratulations from my side and Drug Chemistry Research Center whole team for listed in 2% scientist
Thank you.
in the world. And sir, I have 3 to 4 questions from my
Sure.
colleagues from abroad. Okay. And the first query from Purdue University, Dr. Singh. He is my colleague and he is
Oh,
pursuing,
nice.
yeah, he is pursuing a postdoctoral fellowship. He is asking, sir, are you working on Protex? Because I'm interested in Protex, and what are the most promising current or potential applications of nanotechnology in medicine, and how do they impact healthcare advancements?
In terms of cancer, right now, our lab is working on two things. One is we are working on brain cancer, and the second one is the breast cancer. So, in both of these cases, we are working on different nano drug delivery systems. For example, active targeting is for sure. And we are also working on nanolipid carriers, polymeric nanoparticles, and also on liposomal, different vesicular systems. Now, in this, we are working. One is with the antibodies, one is with the protamine. So all of these studies, because not every study is being possible at our place, we are working together along with Attract. Actually, in Mumbai, we have this Tata Cancer, the hospital and the research center. So there we have collaborations with Dr. Shilpi Dutt, and that is the reason we are able to work so much deep on in the cancer area. Otherwise, we are more focused on drug delivery, nano aspect, and the cancer part of it is being worked at Attract.
Okay, sir. Thank you so much, sir. Now I have one query from South Korea.
Okay.
He is also my colleague. He worked with me in CSIR Triple M Jammu. Now he's pursuing his postdoc. Sir, can you explain how nanomaterials are changing the landscape of material science and engineering, and provide examples of innovative materials that have emerged as a result of nanotechnology research?
Very nice question, and that is what, when you talk about materials. Just to give a very easiest example of PLGA, if you talk about as a material, everyone is aware. All the most of the anti-cancer products cannot be formed without PLGA. But then PLGA alone could have not been possible to reach the correct targeting site. So PLGA then was conjugated with MPAGs. So now in the market, you already have MPAG and PLGA different combinations. And again, then you have different materials which are being used for as a targeting moiety. So actually, without the right material science, the drug delivery itself is not possible. Whether you talk about different antibodies and the different ligand systems which can be attached to the polymers. So for me, for example, these days we are working on protamine. So now protamine also, if I take the low molecular weight, for me is good, but the higher molecular weight, the protamines are not useful. So getting the right molecular weight, getting the right molecular structure of these materials are important for me, and only when I get the right materials, I'll be able to make use of them in the drug delivery system. So material point of view, your question is very right that we have to be exploiting the right materials, and we have to be ensuring that these materials are being used in cancer drug delivery. Even for targeting also, when I talk about, unless until I get my material, because which is one is biocompatible, but then is also be able to reach there and remain in the blood circulation for a longer period of time. Now, when you talk about a pulmonary drug delivery system, because that example we have not taken, so not everything is compatible with the lungs. So when you talk about lungs, you cannot be making use of so many surfactants, you cannot be avoiding. So now, because lung is so critical organ, right material where you're able to make use of the right surface charge also, and in avoiding the different surfactants which can be harmful. And again, ensure, because if you say I give you a drug delivery system which is for a week therapy, so now is that polymer or lipid able to be safe in lungs for a week or a month? Is tough. So then comes the answer is right materials, and the right area that we want to reach is important. So biomaterial we need to be very much careful about.
Thank you so much, sir. Sir, I have another question from India CSIR lab, MPRI Bhopal. And the question is, sir, what role, sir, are you interested to give an example related to nanotechnology, which play in environmental sustainability, such as water purification, pollution remediation, and reducing the environmental impact of various industries?
See, these days, when you talk about this upcoming research, sustainability is one topic which we cannot avoid. And I think even journals now, when we are like publishing, they always talk about is the environmental sustainability comes in every context. So for us, definitely, because I don't work in the areas of water and the other part that you've asked, but then even for us, when we talk about biodegradable materials, for drug delivery system, somewhere the concept of sustainability, we are at least able to give it one tick. So for drug delivery systems also, and I think if I can tell you very fast, there will be a system that when you even submit a paper, the aspect of sustainability has to be marked, whether the paper has got some relationship with sustainability. So yes, we need to be taken care of. So for drug delivery aspect, we are trying, but I don't work on the other areas that you have asked.
Thank you so much, sir. So now I'm just opening and requesting all the professors like Dr. Nilima Goswami ma'am, Misra sir, and Dr. Sivani Saraf ma'am to share your experience with us, and like it will be just a discussion for nanotechnology, because we have very good experienced speakers with us. The sir, I just want to say one thing, that the slides was very good
Right.
and informative. I also perform the same things like you presented, because just you presented the penetration of nanoparticles inside the cell, as well as reaching to the nucleus. Okay, sir. So I have used different type of dyes like rhodamine 6G
Oh,
and rhodamine 123, as well as DAPI and other dyes. So I also performed these things, ROS and MMPs. I go through all these things. I also performed ex vivo experiment on the tumor spheroid.
oh, nice.
Yeah, during my
So are you working
CSIR.
on 3D spheroids also?
No, sir. Right now, I don't have facility here, but
Okay.
I can collaborating with you, collaborating with CSIR Triple M Jammu, because I performed this 3D tumor spheroid at CSIR Triple M Jammu. The scientist name was Dr. Goswami, Dr. Anand Goswami.
Okay. So
He
maybe we want
is
to
senior
do
principal
3D
scientist.
spheroids actually. We want to do this study.
Yes, sir.
So here we have option of Actrite, but they have a long queue.
Okay. So I can discuss with sir regarding your query, and
Sure,
that
sure,
would be
sure. Please
interested
let me
to
know.
collaborate. Okay, sir, that would be more better, I think. So I just opened this session for the discussion for Dr. Mishra sir, Asuni Mishra sir, Goswami ma'am, and Sivani Saraf ma'am, and
Okay,
also request to for a vote of thanks.
okay. So initially, like I will go for the official mode of vote of thanks because the topic is it's a kind of universal topic where there is no limit. It's like as wide as the ocean, and I should say
True.
as articulate you as you can, start beautifying acres of land by planting number of varieties of the flower. So this topic has got that length and that, I think, power or vitality, I should say. So first of all, thank you, Professor Khush Yadav, sir. I think this is first time in any platform we are meeting. I've been working for this particular profession for last 15 years in Department of Pharmacy, Barkatullah University, Bhopal.
Okay.
And the kind of slice and the kind of intensive work you have done in the field of nanotechnology is really remarkable, because people says that researcher is good when they work in a variety of the thing, but I personally believe that the person like you have a tendency to find out the gem by dwelling it consistently, which actually talks about the holistic application of certain technology of current century, like nanotechnology. Though, like as we all know that we like you cannot have every drop of ocean, because it's an open sky where anyone can reach anywhere to find the true treasure. But this is, what do we call it, even in a life, if we can even touch the 10% part of something, then I think this is a success of anyone's researcher life in the consistently world of research and development, which is running so fast, and to maintain the pace is really difficult at this point of time for the budding researcher like me, or probably the great researcher like you people, I should say.
Oh, no, no.
So, thank you very much for this, I should say, rational knowledge which you have given to all the learners today. And I hope that they will definitely utilize whatsoever upcomings and take home messages they got today in this particular conference, and probably they will utilize in their research work. And definitely we soon be having a person among us who will definitely be getting his or her molecule probably registered into
Yes,
this
yes.
FDA for certain kind of cancer therapy. So thank you very much. It was really a kind of I should say a mind-boggling presentation, which actually influence each and every researcher if they have got that depth to understand and that sincerity for the profession. I think that is really important. So in between that, sir, even it's my first time to be with you, and I really got
Yeah,
influenced.
I'm so happy to meet you. I'm blessed to hear so many good words. I'm happy to know Dr. Ashwini Mishra. I think we have not met before, but thanks to Ashutosh, at least now we know each other. We should be connected later on also. I'll ensure that we have a talk also. So I'm
that
happy
would be
for this
that would
connection.
be
Thank
that
you.
would be truly my pleasure if it will happen
Thank you so much.
in future also. So thanks again, sir, and on behalf of the whole team, as Ashutosh is the whistleblower for these
Yeah,
kind
yeah,
of thing, and
Thank you
he
so
is
much.
the charming face which actually
yeah.
covering his face with the smile and putting his
Yes,
head into the intensive research work and consistently working
true,
in
true.
order to uplift the research and development as per his length and as per his control. So the thank you, sir, for it. One point I would like to mention here: there was a question for the water. I think cleanliness by someone from the deceased
Yeah, yeah,
Bhopal
yeah.
RRL.
Yeah.
But like polymers like sodium alginate has been tried earlier in order to separate out the pollutant, because the alginate meshwork of the matrix of the polymer will actually retain the dirt from the water. Then later on, probably 2 to 3 years back, a study was conducted in MANIT Bhopal, MSCT College,
Oh,
where the professor found that the feather of the hen and cocks, the feather can be utilized within a certain cylindrical structure, and the feather has got very good adsorbing property. So if someone is really having hunger to do research in this particular field, then they coated the feather of this peacock, this hen and probably cocks, and by coating the polymer, they may even increase the adsorptive power of the dirt which even come out from the effluents, and that's how they
oh,
can even compare the conventional method of filtering and making the water pure by comparing with these kind of novel inventive approaches. So that could be the proper way, and even if the result will come out in most positive way, then definitely it will be a new mode for those multi-billion companies who are actually working in India for water purification, as well as for converting the dirty water into a usable water. I think he or she could try probably in that mode. There was yes, Monu. If you have to say something, you please
No, no, sir.
continue.
I just have a query. Like you talk about the feather. So is these type of feathers having some chemical compounds or something, some types of proteins? Because these feathers are prepared of some proteinous
Exactly.
material. So maybe they contributed some roles to do these things.
Definitely that the protein is there, as well as the porosity of these feather, which actually is quite better in term of retaining these. So up to certain density, if we fix it into a cylindrical container, and then it is being utilized for the filtration of these kind of dirty water. Actually, I read this 2 to 3 years back when I was actually thinking for some topic for my student. So that's how I came to know, okay, these kind of research. And that particular professor of MSCT, he probably got a very good prize, I should award after his research works. So it is just a notion, because or the student even can go for the online literature search for that thing. He will be definitely getting much more notable information, or probably worthy information, if he will like do the dwelling or probably digging out approach for that particular thing. And there was one more. Yeah, please, Monu, continue.
No, no, sir. You continue.
Okay. And there was one more thing, like probably my senior Dr. Nilima Goswami ma'am has asked about PDPK modeling of pharmaceutical formulation. There is a very good company, Western country-based company, GastroPlus. So GastroPlus is the company which is actually giving the data by only using in silico approaches of 100, 200, even 500 patient data. So they are having some such kind of algorithm where whatever formulation you have developed, if you will feed it as per the format they have, so these gastropus will give you a very good guideline regarding PK, PD, not only guideline, but the outcome of the research, which is actually quite similar to the human. Though, like, it doesn't mean
That's an
that we do,
expensive, I think it's quite
it's
expensive
very
software.
expensive, sir. It's very
Because
expensive.
even
Yeah,
in Bombay, it's not available. Earlier, it was there with BCP. Even now, they don't have it.
exactly, because they have raised even, sir.
I think only with industries they will have.
Only with industries, only with the industries. So that is there, but these kind of softwares are actually there in with the world. And sometimes, what happens is we are not aware about it, but these are the things. Even PKSIM is there. I think if someone want to work on PKSIM, it is there, which is free of cost, and anyone can work on it.
True,
So
true.
these kind of PKPD system are there. And if I'm not wrong, from Sartara, there is one more software which is not completely freely available, but some portion of it is available. Because last to last, when I was into this inspection in Maharashtra, where I met Andhra researcher, and she mentioned me that thing, that this Sartara software is available, some part of it available for the PDPK modeling of your formulations. So these things are there. So the platform which we have got is because of my dear student Ashutosh.
Thank you,
So
sir.
again, like I have to thank him, as like I'm very talkative, even by my energy, you can also understand it.
Yes, sir.
So I think there are people around us. Let me give them chance also, so that they may talk and they may.
Sir, professor should be talkative always, because after because of them, the students will learn more.
Yeah, definitely. Thanks for your
And
generosity.
sir, I want to add one more thing, that some researchers from ISER Bhopal just developed the magnetic based nanoparticles for the purification of water. And I
Oh,
want to tell you one more thing, that I'm also working on the same type of particles for therapeutics activity. So Professor Yadav just presented his work slides on magnetic nanoparticles and the targeting. Okay, sir, and like increasing the temperature of the particles of 40 or more than 40, but 43 degrees Celsius around. So these are I am developing nanoparticles for cancer therapeutics activities, cancer
Oh,
therapeutics
nice.
mostly, and hopefully I'm just submitting in 2 to 3 days. And in November, December, I will get the comments from the reviewers. And for the software, sir, 2 to 3 days back on Thursday or Friday, we had a meeting with a company, UK-based company, CCDC. They are developing softwares for in silico as well as different fields of science. So maybe in the coming months we will sign an MOU with them. It's a big company. It's a Cambridge based company, but they are separated from the CDC, Cambridge database or something, and now they are interested to work with us. And hopefully we all will get some advantage from them, and they will provide some softwares for freely available for in silico MD simulations, machine learning, and PKPD and everything, sir. So they are developing a lot of softwares. And so thank you so much, sir.